Well, everybody, thank you for joining us tonight. We have a great crowd with us, and I'm not surprised by that, because the last time Amy was with us, we had one of the highest viewerships that we've had for any of the Dean's Forums, and tons of hits afterwards. So she's about as popular as Dr. Fauci. We have
our own Dr. Fauci.
What prettier.
So let's just begin with a word of prayer. Direct us, O Lord, in all our doings, that with your most gracious favor, and further us with thy continual help, then in all of our works, begun, continued, and ended in you, we may glorify your holy name, and finally, by your mercy, obtain everlasting life through Jesus Christ our Lord.
Amen.
So we're really lucky to have with us Amy Hudson, and I almost feel sort of that we should be calling her Dr. Hudson or Professor Hudson, that we're lucky to have such an incredibly talented person joining us tonight, and somebody who knows really as much about this as probably anybody else does. And so I've asked her for a couple of things. If she was with us, Amy, I believe it was like in April. It was sort of right when everything started breaking loose. And so what I've asked her is maybe just tell us what we've learned about the virus since then, and then to talk about the efficacy of vaccines and face masking and everything that we're supposed to be doing, and just her take on it. And so I think I'll turn it over to her pretty soon. But what I would ask you to do is I would ask everybody to mute yourselves. And then if you have a question, throw it into the chat. And then I will, so we don't have a lot of people hopping in continually. If you throw it into the chat, I will be happy to pass your question along to Amy.
Looks like Amy's on mute.
Yeah, she is. And then we'll take it from there. So thank you, Amy, for being with us this evening. We really, really appreciate it. It really is a great thrill to have somebody with us from our own congregation who knows so much about what is going on. You may recall that she was lecturing in Japan on viruses right as everything broke loose in January, and was on one of the last planes out of Japan before everything shut down. So she knows about it in theory, and she knows about it in person. So without further ado, Amy, why don't we just turn it over to you and tell us a little bit about what we've learned about the virus since the last time we talked.
Thanks, Kevin. And thanks for giving me this opportunity to talk, because it helps. I mean, the virologists all over the world are supposed to be communicating with the public. And I think that's one of the main problems nationally in this country, is that there's not really great communication. The last time I spoke, I felt kind of guilty because I didn't ask you what your questions were. And I thought that this time I would start by asking you for questions that you'd like to touch on in the next 30 or so minutes. And if you have any questions, either speak up now or put them in the chat. I mean, I have topics that I'd like to talk about, but I would love to hear. I don't want to miss any of your burning questions, because I talked about something that I thought was interesting myself.
Well, I would say, Amy, why don't you take 10 or 15 minutes and just talk about what you want to, and then people can toss their questions into the chat, and I will think we'll have plenty of time to hit everything.
Okay. Okay, I'm going to share my screen. Excuse me. I'm sorry about that. Go to my piece of paper here. And so what I wrote down as topics that I thought were interesting since the last time I talked were masks and what we should be doing. I thought it would be interesting to talk about the new B.1.1.7 variant and whether it's more contagious. I thought you might be interested in mutations and the rates of mutations and how important those are. I've recently done a couple of lectures to medical students and graduate students on the mRNA vaccine. So I am equipped to talk about that. And vitamin D, I thought I'd mention vitamin D briefly, and also the monoclonal antibodies that Trump got that helped him survive. I can also talk about those. Should I ask you to vote, or should I just start at the top?
I think start. I mean, we're here to hear you. So.
Okay. The first thing I'd like to talk about is masks, I guess. There's an article today in the Washington Post that said what we should all be wearing N95 masks. And we should, and we can't right now because apparently this country doesn't have enough material to produce them all. And I know that from a question Kevin asked me earlier, because he writes me anytime, and anytime you have a question, feel free to write to me. There are a couple of different kinds of masks. There's a homemade mask. I don't have any of those. There's these blue surgical masks, which fit, as you know, they're square and they fit loosely. There are these KN95 masks, and these have ear loops, and they are they're thick, and they're mostly made in China, but they're better in theory than the square surgical mask. And then there's an N95 mask. An N95 mask is different from a KN95 because an N95 has two straps, one of which fits over your head. This is upside down, I think. And because it fits over your head, it's much, much tighter than this. I just bought these for $50 on Amazon. I got 50 masks for $50, and they are very superior to these because they fit tighter. The challenge of wearing a mask properly is to try to get it to fit well. Masks, if you are wearing one of those cloth masks, so Sue Cornell gave Jamie a fantastic Riverside High School cloth mask that goes over his ear. It fits really tightly, and I love it. And it's not made of this surgical mask material. This has polypropylene in it, which is much better for not allowing viruses to penetrate. But those cloth masks are fantastic because they fit so well. And so one thing you can do is put one of these surgical masks on with a cloth mask on top of it to keep this nice, there's really high-tech material in this stuff that keeps it tight. When you wear just a mask, like a cloth mask, you are protecting other people, but you're probably at risk when you go into a grocery store. If you want to protect yourself from the cash registers cashiers who have their masks on like this, I think the only strategy you have is to put on one of these synthetic material masks that have filtration capability. I just read in the Washington Post that these have just a square surgical mask has 90% filtration capability if you can get it on tight. So a nice cloth mask over one of those is a good solution. The same is true for these KN95s. When I put this on, and I'll try, the loops are too big for me, and so my glasses fog up, and there's a gap at the bottom, so that's not perfect fit. That means that when I breathe, the air is going to come through the big gaps and not the mask. So I always put another mask, a cloth mask over this as well to try to get it to fit tightly, especially before I go into grocery stores. And this is a new thing. They just came the other day, and they do seem to fit pretty tightly. So that's all I wanted to say about masks. You protect people when you wear them. The most dangerous thing you can do is be inside when someone's speaking, especially in a loud voice, without a mask. When I go to the grocery store and I see the cashiers who have their masks over their mouth, and they're breathing and they're sitting in that spot all day long, everything they breathe, if they are an asymptomatic carrier, is going into the air right around that. I do not choose that lane. Okay.
Amy,
yep.
One thing you told me once, as far as the aerosol effect of the virus, is that it's just like if somebody was wearing perfume or cologne, that if you could smell somebody's perfume or cologne, you would also be getting their COVID bacteria in the air. Was that an accurate assessment?
I think that's a good rule of thumb. And whenever I smell someone's cigarette smoke, I cringe because I have a feeling that their aerosol is coming out and then sitting on the particulate matter that they're breathing out when they smoke as well. So, as far as you can stay away from people, the better. And as I said in the last time I was speaking, I do not want this virus. I know that a lot of people, most people, 16, 70, 80% of people are asymptomatic. They don't even feel it. It's been shown that even in high school, Penn State football players, that some huge percentage of the asymptomatic football players for the Penn State team ended up with myocarditis and heart problems. This virus infects heart, liver, lung, kidney, and brain. And I don't know nobody knows what the effects of this are 10 and 20 years down the line. And I hope that I don't get the opportunity to find out. It really, yeah, okay, so I'll stop there. I've been listening to TWIV, This Week in Virology, the kind that make you sick. It's a podcast by geeky virologists, one from Columbia, one from Gainesville, Florida, and I know them. And they, Vince Racchianello has a podcast, and he has on it a guest, Daniel Griffin, who's an MD who is actually the head of United Healthcare. He's really smart, MD, Ph.D. And he talks once a week for half an hour to the clinicians, and lots of clinicians listen for updates in how we should be treating COVID-19 patients with the infectious disease that coronavirus causes. He said that vitamin D, if you come into the hospital with symptoms of coronavirus infection, and you are low in vitamin D, you have something like a 30% chance of ending up on a ventilator, as opposed to if you come into the hospital with normal high levels or normal to high levels of vitamin D. So this is not a good time to let your vitamin D lapse. Take vitamin D. It can't hurt. And actually it can help. They don't understand at this point why vitamin D helps, but they've done the studies, and it looks like arriving at the hospital with low vitamin D levels is a detractor. Let's see. Does anybody? I could talk about the new variant that we're seeing. It's called B1.1.7, and everybody is.
Amy, just back up 1 second. We have a question. How much vitamin D should you take every day? How many units?
I personally take the lowest amount, 1,000 units. I get them from Trader Joe's. You don't have to be high in vitamin D. You just have to not be low.
Okay.
And we all live in Wisconsin, and we stay inside all the time. But I think whatever unit you can get over the counter. I don't usually take a lot.
I think people are probably also really curious about vaccines, because we have a lot of people in that 65 and older demographic, and some of the people have gotten them, and there seems to be a lot of confusion over how to get in line for them and how efficacious are they. And the Moderna one, first the Pfizer one, and now I just read today that Johnson & Johnson is pretty close to having one approved on a trial basis also.
Mm-hmm. Okay, so that's where I'll go. The vaccines are mRNA vaccines, and the two vaccines are Pfizer and Moderna, and they're basically the same. They both use messenger RNA, and it's encoded in it's called a lipid nanoparticle. And so it looks like this is a lipid nanoparticle around here, and then there's a piece of messenger RNA inside. When we get vaccinated, does anybody know where they make the flu vaccine? Out of John Bendler, I saw you here. Where flu vaccines are made? They are made. This blows my mind. The flu vaccine is made in chicken eggs, and they make 5 million doses of. No, they use 5 million eggs in making a flu vaccine, and they don't just use regular eggs like we get at the store. Those 500 million eggs have to be fertilized eggs, which means that a rooster and a hen have to be together in the hen house at some point. And every single one of those fertilized eggs then gets brought to Merck or whoever makes these vaccines. Sanofi makes them, and and they get injected at the same time. And I'm telling you this because I wanted you to appreciate how what an incredible effort it is for the United States government to have these chicken farms that are under top secret, lock and key. Nobody knows who they are, and they have to be incredibly sterile. You can't let the chickens or the hens get, I don't know, chicken virus that's going around at the barnyard. And then the logistics of getting these eggs into a gigantic pharmaceutical company and keeping everything sterile. They devote about 80% of their money to quality control for these flu vaccines. And the reason I'm talking about this is because it's hugely expensive to do, and it's hugely expensive to get approval to for example, change the vaccine, because you're depending on animals, chickens, to produce the virus. So it turns out that the chorioallantoic membrane of the chicken egg has a lot of cells in it, and those cells are susceptible to influenza virus. You can inject a little bit of the virus into the egg, and then the cells get infected and they churn out tons and tons of flu virus. And then they go into the egg, they harvest the flu virus, and they inactivate it. And that's what our vaccine is. It's just inactivated virus. There's a couple of different kinds of vaccines that you can get. So the flu vaccine is inactivated virus made in chickens, eggs, fertilized. And there's a 60 Minutes you can watch that illustrates this. It's incredible. Mike Wallace. The mRNA vaccines are a little bit different, and they've come to market really fast. And the other thing that I thought was really interesting is that there has never been a licensed mRNA vaccine. This is the first for us. In fact, these aren't even licensed. This is this emergency use authorization that they have. So none of us have ever had an mRNA vaccine before. An mRNA vaccine is different from a typical vaccine in several important ways. One is that in order to make this little strand of mRNA and the lipid sheath, the nanoparticle that goes around it, called a lipid nanoparticle, and this is mRNA, is that you don't need any chickens. And you don't, in other vaccines, this is from John Bendler, other vaccines, you make proteins from viruses. So for example, hepatitis vaccine. The hepatitis virus looks like this, and just like the coronavirus, it has some kind of spike protein. It's called a surface antigen on the top of it. And what we do is we make just this protein here in bacteria, and we get bacteria to produce the protein for us, and then we purify it. And then again, we're reliant on the bacteria to make the protein for us. And that entails tons and tons of quality control, because we're relying on something that is living. For this mRNA vaccine, we can synthesize this piece of mRNA in a tube. We don't need animals, we don't need bacteria, we don't need anything alive. And this lipid particle we can synthesize as well. And for that reason, the regulation and the cost of producing these mRNA vaccines for Pfizer and for Moderna is 10 times less. So the speed with which they could bring this to market was 10 times faster than the speed with which you would have, and the regulations to get by the regulation, the FDA regulations for an influenza vaccine, or if you wanted to change the influenza vaccine, or these, or mostly any other vaccine that we've been given to date.
Amy, does that mean that they could also respond faster to variants and to change the vaccine as things come along that haven't been known prior to that?
It does. It will be much faster to do that. And it's another good question. I wanted to talk, I wonder if I've touched on all of, so we've never had an mRNA vaccine. The only people who've had them are all of the healthcare workers so far. So that's 30,000 people in the original trials for both the Moderna and the Pfizer vaccines, I think. And then now probably 1 million people have been injected with the first round. All the healthcare workers certainly throughout the country have gotten their mRNA vaccine, and they have very, very few adverse reactions. A lot of people get a fever and soreness at the injection site. I actually had the vaccine last week, and I got a fever and soreness. But I get a fever with the flu vaccine. If you get a fever and soreness at the injection site, you're in good shape. You're making an immune response, and that fever is caused by your own immune system. Somebody in church actually one day said, you know, every time I get the flu vaccine, I get sick. I get with the flu, and I'm not getting it anymore. And I said, no, it's not possible. When you get a vaccine, if you get a fever from it, it's not getting the disease. It's not possible because you're not injected with live virus. You are getting an immune response. And if you feel an immune response, that's a good sign. It means you're making a good immune response. If you don't feel an immune response, that's okay too. You're still making an immune response. But if you do feel one, that's evidence that it's working. Anybody with questions?
No, Lynn Miranda noted that she did have a fever when she got the vaccine.
Didn't?
She did. Tired and a mild headache.
Yeah. I didn't have a fever, Kevin.
No, you said tired and a mild headache.
Yeah. How about, have you had the second one yet, Lynn?
No, I have not. I'm a little scared from what I've heard.
Yeah, I would expect one. So I had a friend in Boston who had her second one, and she had a fever the first time, and she had a fever the second time, and she basically stayed home and rested. It's no big deal. It's not like 103-degree fever where it's a mild fever and you're tired and you take a day off. And the older you are, the less likely chance that you will develop these after effects or side effects, because your immune system is, it's older. So I think that, 20 and 30-year-old nurses often develop side effects. And unfortunately, I hear that it's as few as 40 to 50% of nursing home personnel in rural areas and in hospital workers, nurses in rural areas are choosing to have this vaccine. And in fact, my father's hospice caretaker told my mother that she was not going to get the vaccine because her friends got sick. And that's, on us. We're not doing a good enough job, or there's some, information out there that's doing a better job conveying that it's going to make us sick. It won't make us sick. It might give us a fever, but it's much much rather have a low-grade fever than the coronavirus. So far, there haven't been any. There's 1 in 10,000 people get anaphylactic reaction to the polyethylene glycol, which is in this lipid nanoparticle, they think. And no one has died of it. They understand what it is. That's why there's a 15-minute waiting period afterwards. And, they know what to do. They give Benadryl and they give epinephrine, and everyone's been fine so far.
I think I read an article in the Wall Street Journal that said that the odds of getting sick from this vaccine are no more or less than the regular flu vaccine, but it's just getting more publicity because of where we are with COVID. Does that square with your data?
Yeah. I stood in line for it. When you're called, when it's available to you, get it. If you've had coronavirus already, get it. The only people who can't get the coronavirus vaccine, at least for 3 months, are people who have been given those monoclonal antibodies that I talked about earlier that President Trump got to help him get through this the coronavirus. And there are reasons for that, and I'm happy to talk about them if we have time. So some people have said, oh, well, it can change your genetic material, it can change your DNA. It can't. So this is messenger RNA, mRNA. We have cells in us that have a nucleus, and our nucleus houses our DNA. This is mRNA. And what happens is the mRNA gets into the cells, and mRNA is a message. It's messenger RNA, and it codes for proteins. And those proteins get made in the cell, in the cytoplasm. So that mRNA never gets into the nucleus, and it can't interact with or recombine with DNA. It's not going to change anything. It's actually a really clever idea to use mRNA. It makes the vaccine, in principle, different from any of the other vaccines that we get, the ones that are based on protein or the ones that are based on inactivated virus. And I don't think I have enough time to talk about the really cool science behind that, but it makes the mRNA vaccine, in principle, better than a protein composed virus or an inactivated virus because of the type of immune response that it generates. We all have heard about antibodies and neutralizing antibody. I think that's in the news a lot. And it talks about antibodies that we make that will neutralize the virus. In addition to, so most vaccines, the goal is to give it to you, and then for you to be able to generate antibody that will neutralize the virus particle. And I'll talk about what neutralization means. But in addition to an antibody response, we also have an adaptive immune response with cells called T cells. And the mRNA vaccine, in principle, gives it the opportunity to generate both a T cell response and a B cell response or an antibody response. So I'm hoping that the vaccine will give us sort of a two-pronged immune response to this vaccine.
Can I ask you? My dad was hospitalized in July for 10 days with COVID, and so I guess maybe the conventional wisdom might be, since he's had it, then he's good, he's safe, because he's generated these antibodies by virtue of having it. So is that true, or how long does it last? We've heard conversation about how long the antibody reaction lasts and that kind of thing. And in relation, I mean, really vaccines, as I understand it, sort of trigger this antibody response. So what's the difference between a vaccine and actually having it?
Okay, I would think that having it would be better than getting the vaccine. However, that's just what I think. It's not what I know, and I don't think anybody knows. How long does protection last is a really, really good question, especially in how long does protection last in a 92-year-old. If that 92-year-old has the opportunity to get the vaccine, despite the fact that he should have some protection against it, we don't know how long that protection is going to last. And it should be different in a 92-year-old from a 20-year-old. And because we don't know, I think the wisdom is to not pass up the opportunity to get the vaccine as well as your own protection, because you never know you don't know what kind of protection you're going to raise when you have the virus. And that's what Daniel Griffin on, my favorite guy, Daniel Griffin on TWIV is saying to the clinicians who he talks to on his weekly half-hour podcast, is to get the vaccine no matter who you are and whether or not you've had it. In terms of how long the protection lasts, as I said, we don't know, but right now the theory is, the data out there says that as long as 8 months after the vaccine, we're still protected. And we haven't really had much more than that to go on. Everybody thinks that's a really good sign. And so, but we get an influenza virus vaccine every single year. And we get an influenza vaccine every year because the virus mutates. Viruses are known to mutate, and an influenza virus mutates 2 to 4 times faster than coronavirus. We get an influenza virus vaccine every year because every single year the flu virus has mutated away from the protection that we had from the year before. So right off the bat in April, we were saying that because coronavirus mutates 4-ish times slower, that maybe we won't have to have a coronavirus vaccine, maybe the most frequently we'll have to have a coronavirus vaccine is every 3 or 4 years. That's what I'm hoping. It would certainly make life easier. But there are 2 factors that contribute to how often we need a new vaccine. One is the mutation rate, of the virus, and the other is how long and lasting is our immune response. And nobody knows this. We do have an idea of this. So in a perfect world, if we make a great immune response, probably 3 or 4 years, we might need a new one. If our immune response wanes, and there have been papers out there that said it doesn't, for example, there have been people who have gotten coronavirus twice, and presumably they got it the second time along with symptoms because their immune system had waned after just 4 or 5 months. That was really concerning, and that would mean that these vaccines, although they're 95% effective in over a period of X number of months that can't be longer than a year, we might need boosters every year or so. So I don't think we know. I think that the people who study this are optimistic that we won't, that the immunity that we're getting from the vaccine is long lasting. That said, we've heard a lot in the news about this new variant. It's called B1.1.7. And what that is I wrote it down. It's a N501, they call it the N501Y variant. From a virologist perspective, it's not a strain, it's a variant. In the beginning, we kept hearing on the news that there's a new strain out and it's more contagious and it's going to be horrible. It's so far, it's not classified as a strain yet, it's simply a variant. And the difference is, so as I said, these viruses, all viruses mutate, and they mutate faster than we do. I've got a they mutate faster than we do, and they mutate just as because they are they're small and they can make errors. The coronavirus has a proofreader, which contributes to its lower mutation rate than influenza virus. So that's good for us. I just sort of wanted to talk about mutations and talk about what they are and whether they're normal and natural. And when they say that coronavirus N501Y is a new variant, I wanted to sort of remind everybody that a protein is a long string of amino acids. And so I've drawn a long string of circles here. And so the spike protein that is on the surface of the corona that Neil illustrated really well in his 3D printed model, that spike protein right here is just a long chain of amino acids. And when they say N501Y is mutated, then in this chain, the spike protein has 1,273 amino acids in its chain. And so this single amino acid here out of the 1,200 has changed. It's not a lot. So then the question is, now with this new variant, are we still going to be immune? If we have this vaccine, will the vaccine still work? If we had coronavirus, now that this new contagious variant from England or the UK is coming, what if we're not immune anymore? And so when we make an immune response, and I drew this up here, what we make are antibodies. We make antibodies, and those are what protect us. Neutralizing antibodies are what we hope to make. And so, as you can see here, I drew this spike protein on the surface of SARS-CoV, and Neil has a 3D model of this. And that spike protein, it's supposed to interact with this ACE2 receptor on the surface of our cells, and it binds well. What they think is that one single point mutation, N501Y, allows the spike protein to bind more tightly to our ACE2 receptors. We make antibodies, our immune system does, we make antibodies to the spike protein, and that's what could be neutralizing. And what a neutralizing response is when we make antibodies that recognize that spike protein. So that on virus particles, we have antibodies that will coat the surface of the virus. They'll coat all these spikes And with this antibody bound to the spike protein, then this virus can't bind to ACE2 because the antibody's in the way. That's a neutralizing antibody. When we make antibodies, we don't just make antibodies to that particular amino acid. We make antibodies to everywhere on the spike protein, or a lot of different places. All the places that are accessible to the antibody will make. Some of those antibodies, like this one, might be neutralizing. In other words, with this antibody bound, this virus can't infect these cells because the antibody's in the way. But that we can also make other antibodies like this that bind to the side of the spike protein that may not interfere with the binding of this virus to the cells. And they will also be helpful. They might not be neutralizing. My point is we're going to make, is that there are a lot of different antibodies that we could make that will be neutralizing. So for example, if you had an antibody bound here, if you had an antibody body here, and one here, they all might be neutralizing. So just because that single amino acid is changed doesn't mean that we're not going to also have neutralizing antibodies that bind right here that also prevent the virus from getting in. And so the take-home message from that long-winded description is that it's possible that that single point mutation right here, the N501Y, might change the interaction between spike and the virus and make it more contagious. Because if the spike binds to the ACE2 receptor better, chances are it could be more contagious. There might be a neutralizing antibody that happens to recognize that N501Y, and we might lose that neutralizing antibody, but we would probably make other neutralizing antibodies. And so one single point mutation or one single variant might not eradicate the effectiveness of the vaccine. It might take a many viruses.
What you're saying, these new variants may be more contagious, but the vaccine will still be effective in combating them.
Yes, at this point, the data says that the vaccines, both Pfizer, well, Moderna definitely, and I think Pfizer as well, have both published that they will be effective against the vaccine. You still generate, they're still neutralizing epitopes. That said, I think Moderna has already started to make a vaccine that encodes an mRNA that now encodes a tyrosine at this residue rather than the asparagine. So that's N and Y, and it changes the end of the Y. And they might be working out a way to boost if it becomes necessary. It's no big deal to simply synthesize an mRNA that encodes that mutation. I also wanted to talk about, I don't know how much time I have. Okay. These mutations and the evolutionary changes forced by them. So when our flu viruses mutate so fast, they do that they don't think too much, but they do that as a means of escaping our immune systems, because they know if we get the flu and we raise an immune response to it, then next year when the flu comes around, we're not going to be infected and it won't be able to transmit. So evolution has forced influenza virus into an error-prone polymerase. So it makes more of itself, but it makes a lot of errors in hopes of creating a flu virus that will be able to infect us even though we have an immune response. With coronavirus, the change that variant was not caused by evolutionary pressure, because first of all, it's still infecting us and we have not at all generated any kind of herd immunity in our population. Moreover, the vaccines haven't forced the change and antiviral drugs haven't forced the change because nobody's vaccinated yet. So that's just simply that N501Y variant is just simply a mutation or a variant that occurred in the normal life cycle of the coronavirus. We haven't reached a point where evolutionary pressures of vaccines or our antiviral drugs are forcing it into making a better, stronger coronavirus. And I also wanted to point out that when these mutations happen in viruses, they happen all the time. And most of the time, and I could probably come up with a percentage, or John Bendler could, most of the time, those mutations are going to be either deleterious to the virus, they will make it completely ineffective, or they'll be silent and you won't see them. Only occasionally do you get a mutation that actually makes the virus more infectious. And in this case, it seems like the virologists say that they have not proven that this is more contagious, but the epidemiologists say that it's more contagious.
How about, we just have a few minutes left, so I've been making some notes. So tell me, I mean, it seems to me from what you've been saying that wearing masks, and if you have to double up on a surgical mask and a cloth mask, that might be helpful. Social distancing, make sure you're up on your vitamin D, and get the vaccine when you can might be the most practical takeaways from this conversation.
Those are the takeaways, Kevin. Those are the takeaways. Yeah, I can't remember what.
Without going down the rabbit hole of politics, I'm just absolutely flabbergasted that our legislature has passed a resolution to roll back the face mask requirements.
Especially in light of this potentially more contagious virus that's coming out. I mean, they say that London's hospitals have been overwhelmed. And if it is actually more contagious, why would you not do something so simple as wear a mask?
Amy, I don't understand what you mean. I mean, if something is contagious, it's contagious. What do you mean by more contagious?
Well, epidemiologically, and I am a virologist, I don't understand the epidemiological data that's presented. But epidemiologically, they say that if the population in London or in the UK it all has a certain sequence of its spike protein, and then they notice that the UK sequences their virus and we don't really very much as they do, and they noticed that a new variant entered the population, and they can through some mathematical modeling they can say that the rate at which that this new variant spread within the population was faster than it should have spread if it wasn't more contagious. So I don't understand the math behind that, and I can't evaluate the data. Virologists say the best way to test that is to take the two viruses and put them into an experimental model of humans or mice or monkeys, and then look to see whether it really is. And those experiments haven't been done. And they hypothesize that the way it's more contagious is that single point mutation makes this spike protein maybe bind more tightly or bind better to its receptor, and maybe hang on more, and maybe that's how it gets in better. There's an infinite number of ways that it could be more infectious, but that's a hypothesis.
All right. Well, if anybody has any more questions, throw them into the chat. Amy, thank you for being with us. I have to say that you have really put this forward in a very understandable way. That I know this is very sophisticated and complicated microbiological sort of studies that are going on, but I think you've made it all very accessible to us who are listening here to what you have to say, and I'm very appreciative of that. And we really are very fortunate to have you in our congregation, because I think from what you've been saying, I mean, you're sort of reinforcing what we're hearing in the media, but we trust you, and
trust Dr. Fauci.
well,
Everything he says is good.
Fauci too, but to hear it from you, from somebody we know, is very reassuring. So thank you.
Thanks very much for letting me come. And also, Kevin has emailed me a couple of times saying, what's this KN95 mask? And he's asked me questions that I'm absolutely happy to answer any questions that you might have that come up in the media. Just send me an email, and I'm happy to comment on it.
Well,
thank you.
No, thank you for being with us.
Thank
And
you.
we're all looking forward to the time when we're finally on top of this thing with vaccines and herd immunity and everything else that's going to make it possible for us to be together. I have a feeling it's by next fall, we may be able to gather in sort of a way that we have gathered before. It still won't be 100% as to what we are before, but we'll be closer certainly to where we are right now. And if everybody does their part, everybody wears masks and social distances and is careful and is vaccinated at the earliest possible opportunity.
And even after you have the vaccine, keep wearing the mask until people think it's safe otherwise.
Right. So it'll be good for you personally, and it will be good for us as the body of Christ, that we are able to gather together again in a way that I think is really important to how we express our faith. So thank you very much, Amy. Thank you for everybody who has joined us tonight. And we'll throw some of this up on our website tomorrow on our YouTube channel. So if somebody missed it, they'll be able to hear what we had to say. So everybody have a good night. Stay safe, stay healthy, and we'll see you maybe this weekend on Coffee Hour.
Good. Thanks very much, everybody.